All right, so welcome to our next Infogest webinar. My name is André Brodkorb. I'm based here at the Teagasc Food Research Centre in Moorepark, Fermoy in Ireland. This is already our 13th webinar related to topics of food digestion. It is organized by the Infogest network, which is an open scientific network sharing information on food digestion. And we have 3 more webinars in the run-up to the next conference on food digestion here in Cork. Today we are going further down the digestive tract, so in a theme related to the gut microbiome, which is one session in the food digestion conference. So I would like to invite the panelists. I would like to introduce the first panelist as my colleague Linda Giblin. She's a working group leader here in our Infogest network, and Susan Joyce. Susan Joyce is a lecturer in biochemistry and cell biology in UCC, University College Cork, here in Ireland, and she's also an investigator in the Alimentary Pharmabiotic Centre, also here in Cork, and Susan will introduce our speaker.
Okay, so hello everyone, and welcome again to the 13th session in our webinar as part of the Infogest series. I'm very excited to introduce our host, our speaker, Professor Kieran Tuohy from the University of Leeds, to give this webinar. So in terms of this going forward, if you have any questions post this talk, can you pop them into the question and answer chat function, and we'll take it from there once Kieran has finished his presentation. So anyway, Professor Kieran Tuohy is a professor of energy metabolism and microbiome at the School of Food Science and Nutrition, now based at University Leeds, and I believe he took up this post this morning. Kieran is a microbiologist working in the field of nutrition, and his research focuses on how microbiomes, both within the gut and along the food chain, impact on human nutrition, on health, and on disease risk. Now, Kieran has a particular interest in whole plant foods, but also in fermented foods and their bioactive components. And he's really focused on how they modulate the intestinal microbiota for improved host health. Kieran joined the University of Leeds this year. In fact, today was his first day on site. And before that, he led a team of microbiologists, nutritionalists, and bioinformaticians at the Nutrition and Nutrigenomics Unit at Fondazione Edmund Mach in Italy, in Trento in Italy, for the last 10 years. Between 2006 and 2010, he served as a lecturer in food metabolomics in the Department of Food Science and Nutrition at the University of Reading, where he still maintains links. And before that, he worked as a researcher in the laboratory of Professor Glenn Gibson at Reading. Kieran received his PhD from University of Surrey in 2000 under the supervision of Professor Martin Adams and Ian Rowland. He holds an MSc in environmental microbiology from the University of Aberdeen and a BSc in industrial microbiology from University College Dublin, Ireland. I'm professionally associated to Kieran. We have just completed an EU joint programme initiative together, looking at bile acids for health, and I can attest to his great depth of knowledge and his interest and skills in combining cross-disciplinary research in his investigations, and importantly, his openness and generosity as a scientist. And it's for all of these reasons I am delighted to introduce Kieran as today's speaker in the Infogest webinar series. So please turn off your cameras and your mics, pop your questions into the chat, and let's listen to Kieran's webinar.
Hello, my name is Kieran Tuohy. I'm Professor of Energy Metabolism and Microbiome at the School of Food Science and Nutrition, University of Leeds. And today I'd like to give you a talk about how different foods might mediate some of their health-promoting abilities, especially in terms of reducing metabolic disease risk by modulating the gut microbiome and the role in the intrahepatic circulation of bile acids. To start with, I'd like to give you a brief outline of the human gut microbiome, or microbiota. This collection of microorganisms, which comprises about 1,000 different species, is unique to each one of us, and it changes both in time and space along the gastrointestinal tract, in the stomach to the ileum and colon, from the lumen to the mucosal surfaces, but also throughout our lifespans, from infancy to through adulthood and old age. Now, each one of us have our own collection of microorganisms, and this is determined by our mothers at birth, by our genetics, but principally also by our diet. And diet has an important role in shaping both the composition of the microbiota and also the metabolic activities. It's no wonder, therefore, that the gut microbiome has been associated with many of the diet-associated chronic disease states. In fact, the gut microbiome is beginning to be recognized as an organ, a new organ within the human body in its own right, and it interacts at a physiological level with many important factors that play a role in metabolic disease risk. The gut microbiota provides a barrier to invading bacteria, so it provides colonization resistance. It ferments dietary fiber into short-chain fatty acids, which have important cell signaling activities throughout the body, not just in the gut. They play an important role in the transformation of polyphenols into small phenolic acids that then can be absorbed from the intestine, and again play a role in regulating physiology at extraintestinal sites. The gut microbiota plays a very important role in the enterohepatic circulation of bile acids, and I'll concentrate about this in today's talk. They also play an important role in metabolism of amino acids, especially tryptophan and aromatic amino acids. They regulate satiety, energy usage and storage. So the gut adipose tissue axis, the gut brain axis in terms of regulation, food intake, but also impacting on mood, depression, and cognitive function. And of course, the gut microbiome, both as a source of stimulus for the immune system, but also and increasingly recognized as an important regulator of immunometabolism, determines our immune function, both in terms of chronic disease risk, but also our ability to fight off infections. And obviously, these last 2 years, much focus has been paid on how immune function determines who gets chronic or acute severe infections, whereas others manage to fight off infections like coronavirus. In terms of chronic disease risk, people presenting with all these chronic diseases, colon cancer, inflammatory bowel disease, IBS, celiac disease, atopic eczema, obesity, metabolic syndrome, type 2 diabetes, all present with a microbiome that is strikingly different from normal healthy individuals, both in terms of composition, but also, more importantly, in the metabolic output. So this is just a brief schematic to illustrate some of these points. You might imagine a gut microbiome being composed of commensal organisms that play an important role in colonization resistance, inhibiting the invasion of some of these pathogenic organisms, but also breaking down complex dietary components into smaller metabolites than can be absorbed from the intestine. You might also imagine diet being able to stimulate the growth of beneficial organisms, the prebiotic concept, where certain dietary fibers or polyphenols can stimulate the growth of beneficial organisms like the bifidobacteria, but also butyrate-producing bacteria. And this collection then increasing concentrations of compounds or metabolites we see as beneficial, like short-chain fatty acids, acetate, propionate, and butyrate, but also small phenolic acids, things like folate, biotin, conjugate linoleic acids. But also the gut microbiome plays a very important role in the enterohepatic circulation of bile acids. Now, as we all know, bile acids are released into the intestine in response to a fatty meal, but the microbiota are principally responsible for converting hepatic primary bile acids into secondary bile acids. Also, these bile acids, when they're released into the intestine, are conjugated with amino acids, and microbiota plays a very important role in deconjugating these bile acids and altering their hydrophobicity and their ability to be reabsorbed back through the enterohepatic circulation. This metabolic output of the microbiome, if you like, is very important in regulating intestinal permeability and stopping the translocation of inflammatory compounds like lipopolysaccharides, bacterial cell wall components from gram-negative bacteria, but also regulating immune function through Treg, Th1, Th2 balance, and also activities like the neutrophil reactive oxygen burst and the ability of short-chain fatty acids to act as chemoattractants for immune cells to the intestinal tract, the site of likely infection. I mentioned how diet might shape the microbiome and some of these metabolic reactions. In a recent EU project, this was a joint programming initiative project through the Healthy Diet for a Healthy Life ERANET project. One of them called Kabbalah Diet and Health was led by Fondazione MAC, where I worked up until recently. That partners University of Reading, University of Insubria in Varese in Italy, University College Cork, and Susan Joyce was the lead in this. And we had a partner in Israel, Ben Gurion University. And what we wanted to look at was how different dietary components might shape the microbiome and then impact on their ability to modify bile acids in circulation, and then identify these circulating bile acids as biomarkers of health and disease. And what I'd like to do in today's talk is focus on some of these aspects, giving you some examples from the literature, but also some of the data we're generating in Kabbalah Diet and Health. And we know for a long time that foods rich in polyphenols and dietary fibers, red wine as an example, can impact on cardiovascular disease risk. But more recently, we've also seen that these foods can also impact on gut microbiota. So this is an example of a study where they fed red wine, the alkalized red wine, compared to baseline pre-intake, or compared to gin as an alcoholic drink with very low polyphenols. And you can see significant changes in microbial populations, Bifidobacteria, Ergatella, Brevidella, the Bacteroides. All of these are very important fiber fermenting organisms in the gut microbiota, and you can see them stimulated by the red wine or the dealkalized red wine over and above the gin or the baseline levels. On the contrary, you can see how gin stimulates some of the more harmful organisms like the Clostridia within the gut microbiota compared to baseline. In terms of metabolic disease risk, this study showed an improvement in systolic blood pressure, triglycerides, cholesterol, and C-reactive protein in people when they were consuming the red wine or the alkalized red wine, and this was significant compared to baseline and the gin. At the University of Reading, where I worked previously, Jeremy Spencer and Glenn Gibson did this study where they fed high flavanol chocolate drink to people and saw a very similar change in microbiota stimulation in bifidobacteria, the beneficial organisms, and in the microbiota, and also an improvement in markers of metabolic disease risk, in this case, triglycerides and C-reactive protein. It's Julie Lovegrove in the University of Reading. We've recently completed the study, and some of the samples were also analysed in the Kabbalah Diet and Health work. We looked at how 2 polyphenol-rich apples a day for 8 weeks could impact on cholesterol levels in 40 hypercholesteremic subjects, and looked at various parameters linked to gut microbiota, especially bile acids. And we found that in terms of metabolic disease risk, the whole study population showed a significant reduction in total cholesterol, LDL cholesterol, triglycerides, and ICAM, an intestinal molecule linked to cardiovascular disease risk. The effects seem to be stronger in women than in men. They've also saw an improvement in fasting insulin in the women, and VCAM, another adhesion molecule related to arteriosclerosis in the females. In the men, we saw a weaker effect, but still in the right direction. looking at bile acid profiles in serum, we saw that a significant increase in deoxycholic acid, a secondary bile acid, in male subjects post-chronic ingestion, this crossover trial. We then did some mathematical modeling, mixed linear modeling with the female data, and found that circulating levels of lithocholic acid would significantly predict cholesterol levels in the women as they age. On the contrary, we could see that apple intake and glucose or deoxycholic acid would significantly predict the cholesterol lowering effect of the apple intake. These kind of studies have been shown, and mechanistic studies have been supported in animal studies with various high polyphenol, high fiber foods such as camu, a berry from the Amazon, but also blueberry, raspberry, tea brownings, and epigallocatechin-3 gallate, all of them shown to modulate gut microbiota and impact on the intrahepatic circulation of bile acids, typically reducing serum levels of bile acids, especially unconjugated and secondary bile acids, and stimulating the excretion of bile acids in feces. This is one example from Gao et al. 2019, which very nicely illustrated how animals on a high-fat diet compared to a chow diet, so high fiber, supplemented with blueberry extract, could improve metabolic disease risk. So here you see the chow animals and chow-fed blueberry animals showing lower body weight gain over the intervention period, whereas the high-fat fed animals on top in light green showing the highest body weight gain, whereas the high-fat fed blueberry had a significantly lower weight gain over the period. And this was reflected in all the different metabolic parameters, quantities of adipose tissue, especially white adipose tissue, triglycerides in the plasma and liver, liver enzymes related to metabolic disease risk, energy, metabolism, or thermogenesis over the course of the study, being higher in the animals fed blueberry or the chow diets compared to the high-fat diet. In terms of intestinal permeability, you also saw a very similar profile of activity with the high-fat fed animals showing classical intestinal permeability, translocation of LPS, and increasing markers of inflammation, TLR4, TNF-alpha, IL-6, all being increased in the ileum, colon, liver, and white adipose tissue of the high-fat fed animals. This being markedly reduced in the chow-fed animals, but also the blueberry in the high-fat setting, reducing these markers of intestinal permeability and associated systemic inflammation induced by lipopolysaccharide and metabolic disease risk that then goes associated with this metabolic endotoxemia, if you like. In terms of bile acid metabolism and microbiota metabolism, you could see a striking impact both in terms of alpha diversity, the Shannon index, and observed species in the high-fat fed animals being much lower compared to the control cow-fed animals or the high-fat fed blueberry animals. And this was also reflected in absolute abundance, which is not often measured, but also important in parameter in studying gut microbiota, being lowered by the high-fat and then recovered by the blueberry in the high-fat setting compared to the cow-fed animals. If we looked at what species characterized the different diets, the high-fat was associated with Clostridia, with Ruminococcus, with Biophila. All of these organisms thought of as harmful organisms, whereas the chow-fed animals were associated with fiber degraders, with Bacteroidetes, but also with Akkermansia, which was associated with a lean type profile or anti-obesity type microbiome. And the blueberry fed, high-fat fed animals You could also see an increase in Akkermansia, and bifidobacteria, and Blautia. So these are organisms that we see as beneficial, protective against metabolic disease and obesity, and associated with butyrate and short-chain fatty acid productions. In terms of bile acid metabolism, you can see here the serum levels of bile acids, levels and composition changed drastically between the chow-fed animals and the high-fat-fed animals, with much higher bile acids systemically in the high-fat-fed animals, and it's been recovered when the high-fat-fed animals were given blueberries. This was also seen in different in the genes responsible for primary bile acid synthesis in the liver, but also the ratio of primary to secondary bile acids conjugated to unconjugated bile acids. So the blueberries basically recovering the enterohepatic circulation of bile acids to a profile more similar of animals on a chow-fed diet when they're supplemented in a high-fat setting. You can also see this impacting on bile acid receptors, gene expression of bile acid receptors in the liver, in lipogenesis and lipolysis in the liver. So this was in normal animals fed a high-fat diet or a chow diet and supplemented with blueberries. How about in models of diabetes? This is the C57 BLDBDB animal model of type 2 diabetes. So they're fed a chow diet, but this time supplemented with blueberries. And you can see that many of the markers of metabolic disease risk are improved in the diabetic animals when they're fed blueberries, including body weight gain over the experimental period, but also things like glucose, triglycerides, cholesterol, leptin levels, LDL cholesterol, glucose tolerance tests, insulin tolerance tests, but also markers of intestinal permeability and metabolic endotoxemia in these animals. Looking at the microbiota changes again, the blueberry stimulates Akkermansia, lactobacillus, whereas the DB animals have microbiota more associated with disease-type profiles, with obesity, so the Ruminococcus, the Sulfovibrio, and Rickettsia. The blueberry reduced serum levels of bile acids, improved the ratio of primary to secondary bile acids, and conjugated to unconjugated bile acids. And although not significantly significant, we had a trend towards a higher excretion of bile acids and an improvement in cholesterol metabolism markers. So how about probiotics? Another way of modulating the gut microbiota is introducing live microorganisms in food, probiotic organisms, typically lactobacillus or bifidobacteria. So what happens in the intestine, especially the small intestine where the bile acids are released, when we supplement with probiotics? So the first thing maybe to say is that the small intestine is colonized by facultative anaerobes, by organisms like lactobacilli, streptococci, some bifidobacteria, especially distally in the small intestine towards the colon, where bile acids are released. And these organisms typically reach populations of about 10 to the power 7 or 10 million per gram of contents. This is in contrast to the colon, where you'd expect 10 to the power 11 coloniform units per gram of contents, a much lower density of microorganisms. It's also a very relevant number when you compare it to the amount of organisms you might consume with a typical probiotic drink. So typically, probiotics are consumed at about 10 to the power 9, so 100 times more organisms, viable organisms, that colonize per gram of small intestinal contents. So very quickly, when you're consuming a probiotic this level, this organism becomes the microbiota of the small intestine, and its biology, its genetic potential, its metabolism becomes the metabolism of the small intestinal microbiota, because they outnumber them by maybe 100 to 1. This was very neatly illustrated by Susan Joyce and Cormac in Cork, where they did some very neat experiments with germ-free animals and with E. coli with bile salt hydrolase genes collected from different lactobacilli which inhabit the intestinal tract. So here, what they did was they took germ-free animals or conventional animals and then fed them with either E. coli or E. coli cloned with two different bile salt hydrolase genes and compared them to conventional animals with a replete normal mouse microbiome. And I don't expect you to look at all the different genes that are expressed in the ileum and the liver, but just look at the color changes. And you can see the difference that adding one bile salt hydrolase genes from two different lactobacilli can make in terms of gene expression in the ileum and the liver of these animals, compared to germ-free E. coli by itself without the bile salt hydrolase from the lactobacilli. Between the different lactobacilli with two different variants of the same bile salt hydrolase gene enzyme activity, and compared to conventional animals. Now, bile salt hydrolase is very interesting because it's a gateway enzyme to the conversion of primary to secondary bile salts by the gut microbiota. And when they release these unconjugated bile acids to be metabolized in the colon, you get a range of different bile acid species, which then have different signaling potentials, both in the intestine and then when they're absorbed into the liver and systemically. And these different bile acid species have the potential to activate different nuclear receptors, FXR, VDR, but also TGR5, and these receptors play an important role in glucose, cholesterol metabolism in the liver. They regulate thermogenesis in adipose tissue. They regulate glucose homeostasis, but also the synthesis of bile acids. But this is an example of a human study where they selected the bile salt hydrolase active Lactobacillus reuteri strain and fed it to 127 people that were hypocholesteremic and saw about a 10% reduction in total cholesterol, LDL cholesterol, an improvement in LDL to HDL cholesterol, and so a reduction in absorption of non-cholesterol pansterols as a marker of cholesterol absorption. And so they proposed this as a possible mechanism by how this probiotic could be responsible for lowering cholesterol levels in humans. Importantly, they repeated this in a second human dietary intervention, which is quite rare in the probiotics field. As part of the Valid Diet and Health study I was talking about earlier on, we wanted to explore this a bit further and looked at how this particular probiotic strain would impact on circulating bile acid levels, both in a chronic setting, before and after a 8-week dietary intervention, but also in the acute setting. And we gave people a high-fat meal before and after modulating their microbiota with the probiotic, but also another group with oats with 2 apples a day, same apples we used in the previous apple study, but a controlled diet of cornflakes plus or minus the probiotics in our placebo capsule. And although we didn't see any impact on fasting bile acid levels in serum in the chronic setting, we did see differences in the acute setting. So immediately we saw in the acute setting, before the long-term dietary interventions, we saw an increase in unconjugated bile acids in response to the probiotic ingestion at the same time as the high-fatty meal. This was augmented when we're feeding the probiotic for 8-week dietary intervention. So we saw an improvement in unconjugated bile acids, the Cmax of bile acids released after during the postprandial period but also in secondary bile acids and hydrophobic bile acids with probiotic ingestion. Another part of the Giblin Diet and Health project was to look at bile acid levels in serum as well as polyphenols collected by this direct plus dietary intervention, which was carried out by a collaborator at Ben Gurion University in Israel. The PI was Professor Ira Shai. So they had this ongoing dietary intervention where they intervened with three different dietary regimes. The first was physical activity plus a Mediterranean diet provided by diet, but also provided with typical foods high in polyphenols and fiber, up to about 400 milligrams per day of polyphenols, which was a typical Mediterranean diet level of polyphenols. Physical activity, the Mediterranean diet, and supplemented with green, with extra polyphenols in terms of green tea, mankai, which is a duckweed product they selected and brought to the market as a supplement or an ingredient to use in processed foods that's rich in polyphenols and also in amino acids as a meat substitute, and walnuts. And they were able to bring the polyphenol levels up to 1,320 milligrams per day. Now, they intervened in pre-diabetics over a 6-week dietary intervention with a 6-month dietary intervention with a 12-month follow-up, and there were roughly about 100 individuals per group. They measured many different physiological parameters. We in Fondazione MAC were involved in measuring bile acids in plasma, in circulating bile acids, but also in characterizing gut microbiota with 16S. So the dietary intervention itself was a success. They were able to complete 89, 84, and 91 individuals, mainly males, because of the setting. This was carried out in a military base in Israel. They completed the study. They saw significant improvement in weight loss over the 6 months intervention, especially with the med diet supplemented with the extra polyphenols, so the green med diet, but also with your standard Mediterranean diet, and even an improvement with dietary advice and physical activity. Now, these improvements in weight were reflected in many of the other metabolic parameters that haven't been published. Liver fat in particular was reduced. You can see here the MRI scans, and this was significantly associated with polyphenol content of the diet. This work has been recently published in Gut. We also saw an improvement in bile acids and gut microbiota, and slowly these papers are coming out in press as we speak. Now, this isn't a chocolate drink I was talking about earlier. This is something very different. This is autologous fecal microbiome transplant. And with a subgroup of this population, they took 30 people from each of the arms of the study and intervened with either autologous fecal microbiome transplant or a placebo for 14 weeks following the 6-month dietary intervention. So they took the microbiome at the nether of weight loss. They freeze-dried it, encapsulated it, and fed this microbiome back to the subjects for the following 14 weeks to see could the impact on weight regain. Because as you know, when obese people lose weight on a diet, especially low-calorie diets, they tend not to be able to keep the weight off in the long term. In fact, they rebound and sometimes put on more weight than they had before dieting was even started. So what happened in this case, after consuming the autologous fecal microbiome transplant, 100 capsules, they were able to prevent the weight regain in people who had the fecal microbiome transplant. They were able to prevent the circumference regain, if you like. but importantly also they were able to prevent the insulin rebound. So they were able to prevent people rebounding into type 2 diabetes and reverting to unhealthy metabolic disease risk profile in the long term over the following 14 months. So finally, I'd like to thank Infogest for organising this event and inviting me to speak, for Alan Mackey especially for organising this, and Susan and André for chairing the session and putting this all together. And happy to take any questions. Thank you very much for your attention.
Okay, so thank you very much, Kieran, for that wonderful, expansive and indeed very detailed and clear piece of work, and very exciting results. So I guess I have a few questions, and there are many that are coming through, so keep them coming through on the chat at the same time. But in terms of this, one of these questions is directed towards your green med diet. And that, I mean, that seems to be an amazing diet in terms of reduction and redistribution of lipid. Can you comment on the microbial connections there and what the expanse may be in terms of the what this green med diet is bringing to the table?
Yeah, we looked at how the dietics have modulated the microbiota, and we could see significant change in microbial composition before and after the diet. But we also focused on two classes of microbial metabolites, the bile acids and the polyphenols. The polyphenols, especially microbial metabolites, 2,5-dihydroxy benzoic acid and narangenin, and total polyphenols were significantly correlated with the reduction in hepatic fat in the liver. We've also got data coming out relating total antioxidant or total polyphenol in blood with a reduction in visceral fat in those individuals. And also the changes in, along with the microbiota changes, we also saw changes in bile acids. This hasn't published yet, but they were also correlated with things like changes in cholesterol and insulin sensitivity in particular. So it looks like the diet changed the microbiota, and the microbiome metabolic flux was different going back into the body and then mediating some of these health effects.
Okay, that really is exciting, and I'm looking forward to seeing what comes out of the bile acid data in particular in terms of signaling. But it brings me back to the differential that you see in terms of the polyphenols when you look at males and females. And you mentioned that in terms of this, that in males that the secondary bile acid DCA seems to be enriched in the males, but when you correlate in terms of the females, particularly with lipid levels, that you see this significant change or correlation with LCA and with cholesterol. And just looking at that, it seems really interesting in terms of this gender difference and effect. And do you want to comment on that, or maybe talk about that in context of the signaling pathways, or whichever way you want to talk about it?
Yeah, okay. Well, first of all, our study wasn't powered to look at sex effects. And this is a common problem with the literature, that many of these observations are after the fact. So the studies aren't designed to look at this kind of an effect. Saying that, we did observe it, so there does seem to be something there and consistently there. We see similar evidence in some animal studies. In the human studies, a confounding factor might be the menopause as well, and what happens bile acid metabolism after the menopause and later life. And many studies don't differentiate or don't recruit particularly younger women or older women, it's males or females. And so these factors haven't really been taken into account in human studies. In animal studies, typically you do experiments either on male or female animals, and very few are done with both. So then it's quite difficult to see evidence of this. What you can say is that there is quite a lot of contradictory studies in the literature. So that it might be related to that.
Yeah, sorry, I'm just going to ask one more because it's related to this theme. And this is that you brought up the points that you have this prandial and postprandial effect that were very different depending on whether you use the probiotic or the polyphenols or the different treatments you brought in. And in terms of that, Kieran, and this is a commentary because I think this is something that would be important going forward, including for digestion models, is that there's such a big difference in what we can observe from what's happening through digestion and then later on in the postprandial state. And in terms of bile acids, where do you think there's the most powerful effect, or is that they just have different functionalities if we're looking at postprandial through a fed effect and then later on when we're heading into a fasting stage?
Yeah, I think the postprandial tool is really important for the bile acids. If you look at fasting samples, it's very difficult to go to pick up consistent patterns in the studies and the literature. The intrahepatic circulation circulates 7 times a day. There's a diurnal pattern. There's a lot of confounding issues. So measuring an individual's response with a postprandial challenge is a very powerful way to look at how an individual responds to a fatty meal, but also looking at the profile of bile acids that are induced from that microbiome, especially in the small intestine. So I think going forward, this combination of chronic and postprandial challenge tests would be very useful for the bile acid field, but also for things like polyphenols. Again, fasted samples, we only pick up the remaining polyphenols that are not cleared from the blood fairly rapidly after a meal. So to really understand the kinetics of how food is metabolized by the gut, but including the microbiome, postprandial challenge tests are really useful. Combined with a chronic, then you can change the microbiome. Maybe we weren't as successful as we'd wished in Cabela Diet and Health, but maybe other settings with larger groups or patients with a more radically diverse microbiome that can be changed through diet, then you could maybe pick out some of these disease-associated patterns a bit better.
Okay, Kieran, I'll hand you over to Linda now for a few minutes. I see she's chomping at the bit to get some questions.
So, Kieran, that was a great talk. Thank you very much. So one of the questions here is, it's a broad question actually on the Q&A, is mice microbiota versus human microbiota. Can you just discuss the advantages of using mice and obviously then maybe the limitations that scientists who are getting into this area need to be aware of?
It's a very good and an important point that the microbiota of animal models are very different from humans. Rodents in particular, because not only are the species and organisms present different, but they're also coprophages. So they circulate their own microbiomes constantly by eating fecal pellets. So it's a very different system. Saying that they provide a different insight than is possible with human studies. So because we're dealing with food, it's appropriate that we do human studies to do the cause and effect. Do apples lower cholesterol, for example? But to give the mechanistic insight, we really have to go back into the animals then and follow the flux of metabolites from the intestine into the different tissues, looking at what those metabolites then do in terms of gene expression or different physiological functions around the body. We can't do that yet in humans. And so going that way, starting with the human studies, and then doing bespoke animal studies to look at mechanism, I think is still very powerful, despite the changes, the differences in microbiome.
I would agree. I think it's a great idea to take the human study first, where you've got an effect, and then work back into your animal study rather than the other way around. I think it's a good way of looking at it. The other question that's coming up there is about the gut barrier. So you talked about polyphenols and blueberry and stuff like that. Do you think that they have a direct effect on the gut barrier, or do you think it's through the microbiota?
The antioxidant potential of these polyphenols will be much greater in the intestine than there will be in the blood. So in the intestine, you're talking about millimolar concentrations. In the blood, it's at most micromolar concentrations. So there will be a much greater antioxidant potential. Oxidative damage is probably one of the main inflammatory responses and causes leakiness of the intestinal tract. So there is a mechanism there. Of course, this is all within the context of a microbiome, stimulating the immune system, triggering inflammation. So I think they're working in concert. You're providing a beneficial microbiome, providing short-chain fatty acids that regulate expression of tight junction proteins, that stimulate mucin production, that harvest mucin. So Akkermansia grows on mucin, it eats mucin and stimulates the production of more mucin. So this all builds for a healthy gut barrier. And then the antioxidant potential of polyphenols on top of that, relieving oxidative damage and as a result of inflammation. So the two working together, I think, would be the way I see it working.
Okay, that's great. So André, I might hand it back to you, and thank you very much again, Kieran.
I do have one question, Kieran. First of all, thank you for a very nice talk. I do have a question. So a lot of us working in food digestion, we work with in vitro models. Is there space for in vitro models in your field of work when it comes to bile assays and bile acids?
Yeah, but I've done a lot of work with in vitro models, especially with the model that Glenn Gibson developed in Cambridge before he went to the University of Reading. Gut models are very useful at finding out what's possible within for the microbiome. So you won't, like the animal models, it is very difficult to reproduce exactly the same composition of the microbiome. You will lose certain species, but the core functions like bile acid metabolism will be retained within the in vitro models. So then you can look at how different food components or dietary factors can shape the microbiome and then shape their potential to modify bile acid metabolism. One thing we did in from That's Only Mac before I left, we were looking at polyphenol metabolism or fermented foods, and then taking the supernatant when we, for example, use a sourdough through a gut microbiota in vitro model and looking at how the metabolites then impact on cell lines in terms of intestinal barrier function or immune response. We're co-culturing intestinal cells with PBMC and looking at inflammatory pathways. So you can do fairly nice mechanistic studies to show what's possible. But like the animal studies, when we're dealing with food, it's important to do the human studies, and probably the human studies first, and then the in vitro and the animal models to provide the mechanistic insight after that, once you know it's working.
Very good, yeah, very comprehensive answer. So thank you again, Kieran, very nice talk. Thanks for coming. I know you just started in Leeds, so it's a bit of a challenge. But thanks again for agreeing, and also Susan and Linda, thanks for being on the panel. I just have a few more minutes. It's just a couple of slides that I want to show you on the Infogest network, on the conference, and what the next webinars are. I do want to thank Muireann Egan, who is always in the background, but she's organizing all these webinars, and without Muireann there wouldn't be any webinar. Okay, thank you, Muireann. Okay, I'm just sharing my screen there. Give me a second. Can you see my screen? I think you can, yeah. So the Infogest network came out of a cost network a number of years ago, but it's still fairly active. It is free to join. It's very, very open. We have 6 working groups, and we usually meet at the food digestion conference every 2 years, but there hasn't been a conference in a while. So this year we are really looking forward to their conference, to meet each other in person. So there's 6 working groups, and they really cover the whole food digestion area. So from models, food interaction with meals, meal digestion, absorption models, where Linda Giblin is active, the lipase and lipid digestion, starch digestion, amylase in silico models. There's no working group on gut microbiota. So if you have a volunteer, you're welcome, Kieran. There's a new challenge. Okay. We have an email list, okay, but we also go through social media. So if you want to join, send an email to Nathalie Lamar or Didier Dupont. If you don't get to write down the email, don't panic, because we have this webinar on YouTube and will stay there for the foreseeable future. We have a LinkedIn group which I administer. So if you go to LinkedIn and type Infogest, and Infogest stands for sharing information on food digestion. We currently have 1,000 followers, and if you have a paper that you would like these 1,000 followers to see, all you have to do is when you post something at Infogest, not hashtag, but at Infogest, and it's a very easy way of communicating. We also have a Twitter channel. I mentioned the YouTube earlier on, so if you go to YouTube, just go to look for Infogest, and there are all these different webinars on the site already. And at the moment we are broadcasting live. So I have my phone right here. So we are broadcasting live, and we have a number of people on YouTube following this webinar live. And I hand over to the chief organizer of the Food Digestion Conference, that's Linda.
So yes, indeed, we're having our 7th International Conference on Food Digestion in Cork in Ireland in May. It's 3 years since we last met because of the pandemic. So we'd like to welcome all of you to come to Cork in May. Now, the abstract deadline has passed at this stage, so we want to thank everyone who submitted abstracts. Certainly the quality of the science is excellent so far, and we're reviewing them at the moment. The early bird registration closes on the 17th of March, so remember that for all of your calendars. So if you want to switch to the next slide, André, I think. So these are all of the speakers that have confirmed. So we do have a session on gut microbiota. It is session number 5. So the 5 sessions are food structure and food digestion, food processing, food digestion and bioactivity, digestion models in vitro, in vivo, and in silico, and of course the effect of food on gut microbiota. We have, there are all of our speakers there: Tara Growett, Avi Spiegelman, Fiona Gribble, Suzanne Hutchinson, Karen Scott, and Paul Cotter are our speakers for the next International Conference on Food Digestion. I do want to say before I finish that I would ask everybody to check out the website. There are links there to the government guidelines for COVID, for going to the hotels and for arriving into Ireland. So please keep an eye on these. I did a little video about 2 weeks ago, and it's already out of date. So just keep an eye on the website and click on the Irish government guidelines. So be familiar with them before you travel. Now, if you have any questions coming up to it regarding that or anything else, use the email website, because that's manned all the time and is there to answer your questions about the conference. So you can see the email is up there at the top. But other than that, we are really looking forward to seeing you in Cork in May. So hand it back to you, André.
Excellent. So we have a website, and we went mad on social media as well. So on LinkedIn, we have a event, so you can just click on that and get news. And we also have a Twitter account, hashtag ICFD, so International Conference on Food Digestion 2022. You can follow it and they get news. So we have, in a run-up to the conference, we have 2 more webinars, and again, excellent speakers. The first one is Luca Marciani from Nottingham, and he will be talking about MRI, food digestion and MRI, really cutting-edge research. And our last webinar, at least for the foreseeable future, will be Paul Mohan from Massey University in New Zealand. And that will be very interesting webinar on food sustainability and proteins. Debunking the myth is a very interesting take on sustainability and especially the protein digestibility. So until then, thanks for turning up and thanks for your attention. Goodbye and slán.